So it's now time for our panel discussion, in which we will invite all of this morning's speakers to join us on the stage just as soon as the seats get set up. Just while that's happening, I just wanted to point out that we have received approximately 100 questions during the course of the morning. So I'm thrilled to see the enthusiasm and that there are a lot of really excellent questions to get through. For the purposes of trying to get through as many as we can, I am going to focus mostly on the questions related to the research, directly related to the research that was presented by our speakers this morning, as there are just so many, and these are the types of questions that our panel is, of course, most equipped to answer. And of course, they're invited to make any more general comments on related areas as they see fit, but I'll focus the questions that way for the sake of efficiency. I do also want to point out that there have been a lot of questions about specific treatments to get recommendations. This is a research-focused symposium, so as such, we cannot really provide advice on which treatments you should be taking. The questions in the panel discussions here are going to be focused on what the research is telling us and the routes towards developing better treatments for everybody, but for more specific advice, we would of course recommend that you speak with your physician. Okay, so with that, I'm going to invite our morning speakers to join me here on these chairs. Thank you. So we saw a lot of things this morning about the different sample types that people are using for their research, and there were some studies with cerebrospinal fluid, CSF, not to be confused with CFS, blood samples, serum samples, plasma samples, urine samples. I wondered if we might start off our discussion by just talking a little bit about what the different things are you can learn from those kinds of samples, why you might want to try certain ones for certain kinds of measurements, and what you hope to learn that way. So I invite anybody who has a comment on that to, Jonas would be a great person to start. Yes.
Thank you very much. Then I can promote my favorite body fluid first, I guess. The reason why, I mean, of course, you have to consider what is obtainable and what is the situation where we can actually gather material. And when we look for, and also what the reason for the research is are we looking for a new biomarker for something? If we can target the tissue of interest as close as possible, like if we can even get pieces of that selected tissue, it's fantastic, but very seldom we can do that. So in my case, we are very much interested in cerebrospinal fluid, which is as close as you can get to the central nervous system without actually taking biopsy material from that tissue. And it actually reflects, the cerebrospinal fluid reflects the chemistry that goes on in the central nervous system in a very delicate way. However, it is invasive, and I know that many patients won't tolerate to do a lumbar puncture, for instance. So then, of course, we have to step out a bit further out, and then we, of course, target something more easily obtainable, like plasma or serum samples.
Any other comments on that?
I would say, for the same reason that Jonas evoked, plasma is probably more useful, and I prefer I find that plasma is also more sensitive than serum for many molecules, whatever you are looking for. So overall, we have to eventually think about something more practical for the patients and clinicians. So I think plasma is my first choice.
Do you want to maybe explain for people who don't know what the difference between plasma and serum is?
Serum is when you let the blood coagulate by itself, and you have the supernatant so that will be the fluid that will that will be going out from the cells, if you wish, as opposed to when you centrifuge and you obtain the plasma. So that's why the plasma is more concentrated in many metabolites or many proteins or molecules that you like. So
there was a recent study in PLOS ONE who did a formal comparison of metabolite patterns in serum and plasma. And turned out that both were not exactly the same results, but both were useful. I could be.
When it comes to proteins, we know that plasma and serum. In protein case, plasma is to prefer because the coagulation mechanism that we rely on when we produce serum is a bit unreliable. It will change, and the rate of coagulation will change within minutes in the individual. So that's why we prefer plasma if we can.
Okay. All right, thank you. So I'd like to move on to the topic of diagnosis. That's something that really came up, of course, in Ron's opening remarks, but it figured very prominently into our discussions in the last 3 days with the scientific group. And clearly diagnosis needs to improve. I don't think there's anybody who disagrees with that, although we did have a very spirited discussion between our engineers and our physicians regarding how specific and quantitative the parameters for diagnosis should be. And I think the consensus is we certainly want to improve the quantitative and objective nature of the way that ME/CFS is diagnosed. We've got a comment from a patient saying that the doctors say there is no specific diagnostic test for it. They got diagnosed based on a questionnaire and recommended CBT, cognitive behavioral therapy, and then there was a question about what. Yes, there was a question about what kinds of tests can be done to prove that something is wrong. And I think that's an excellent question, and that's something that all of us want to know the answer to. We're all working to find the answer to. So I wanted to invite the panel to discuss how their research figures into the plans to discover better biomarkers based on the biology of the disease, not based on questionnaires, objective measurements, and extending that beyond sort of the studies that have been done so far. How big cohorts do we need to get into to be able to do this? What sorts of biofluids? We've alluded to some of this, but specifically with the goal of finding an effective biomarker and quickly. What would you say?
I actually have a mic. I have a mic, so I don't need one. I would say that we're not ready to use metabolites for analysis, given all of the variables that can be causing why your metabolites are different. One thing I will say, though, is that every time we have a finding that the patients are really different from the controls, I'm hoping that will be chipping away at the idiotic idea that this isn't a real disease. So even if we can't use something for that's not ready really for use for diagnosis, it's still evidence that objective evidence that people are very different than healthy people. People who have the disease are very different than healthy people. As I said, I don't think we're there yet with metabolites. To really have a diagnostic test, you would want to make sure that there aren't the same differences in other diseases, and we're not there yet.
Other comments?
And I agree, and I also think that for most of the differences that we see in metabolite patterns, for the single analysis, they are usually within the normal range, but there's a difference at the group level, but with extensive overlap. And to have a diagnostic marker, you would need much more sensitivity and specificity. Mm-hmm. And what we can see, but as Maureen said, and also in our data, that there's no subgroup that's distinctive from these analyses that tells us that there probably is a feature about the disease. So maybe when we work further along that route, we can see other markers that have more sensitivity and specificity.
Mm-hmm. Can you talk about the difference between sensitivity and specificity?
It has to do with in the detection of false positives or false negatives. How often a test can give a positive result, but it's not indicated of the disease, or vice versa. Yeah.
Okay, thank you.
I would say that one important thing is because the patients are at different disease stages, different triggers, they are using different medications, so we need to apply whatever the test of the choice at a common challenge that will be standardized. So it could be the test like Maureen is using, could be something maybe less invasive and portable that you can visit the patient or the most severely affected patient. But at the end of the day, we need to neutralize as much as we can other things that can interfere with your favorite biomarker at some point, that when the patient becomes his own control. And of course, you have to establish the reference value testing the normal population. So doing that might explain why we started to see, using circulating microRNA, that we start to be able to stratify for the very first time patients. And you saw the symptom of the patient, and now you can revisit other molecules that can explain the symptom. And if you can explain the symptom, the question is it just purely an association, or there can start to be some causal effect. And if it's causal effect, you can go on and eventually try to have more rationale to establish a clinical trial by targeting something very specific that can improve one or two symptoms, maybe not all of them. So that's why I think applying a challenge, and that could be also an in vitro challenge using the cells of the patient, like the group of engineers are working also. So you can do it on the patients. I would say that's the ideal situation in some circumstances. But also you can extract the cell of the patient and apply a challenge in vitro using whatever the test to stratify the patients. I think that's the key that will help us to move forward as fast as possible.
I would like to add to that also the importance of having methods that can give absolute quantitative analysis, because if you have absolute quantitation, let's say nanogram per ml of plasma, then you can translate that to other laboratories around the world. It's a global information, and then we can also allow for comparison between different technologies and methods. So that's something we have to do, relative measurements at some point, but we need to do this absolute quantitation in the end.
Yeah, I agree with what was said, and just like to add that when we try to identify biomarkers, we basically try to use it for diagnostics, to guide therapy, and to monitor the response to drug therapy, which was talked about earlier. So I don't think we really are there yet for any one of these tasks, but I was really encouraged by Maureen's talk that at least from metabolite side, different studies seem to show similarities in terms of the difference between patients and controls. I think we need to do a lot toward comparing different studies and try to get a consistent result that might be used as biomarkers at some time.
That's maybe a good point to transition into a discussion about metabolism, which was a pretty common theme throughout the talks this morning. I wonder, so Oystein, you mentioned that your observations suggest an ineffective utilization of glucose in the patients, and that there are these compensatory mechanisms for the ineffective utilization that actually differ across the sexes. So that was actually a popular sort of a point, and I wonder if you might be able to comment on how these sex-specific differences how the sex-specific differences between males and females may cause the disease to look different in males and females.
I am not sure if I can answer that in detail, but I think that what we have tried to do is look at the data and see how can this fit, and put it together with other data and see does this make sense. And I think that the underlying abnormality is probably the same in men and in women, but if that thought is true, that the patient tried to cope for an inefficient perhaps glucose utilization, and they use alternative substrates for keeping the energy level up, that men and women use that a little bit different. And we think that it looks like both sexes use fatty acids, which are lower, at least in serum, in many of them, but that male use maybe muscle catabolism to a larger extent than women, while women draw from the serum pools amino acids that they can use for this purpose, and also fatty acids. So if that is enough to give a different clinical picture, I don't know. I can't answer that.
Okay. You had mentioned that certain amino acids and fatty acids look to be bypassing pyruvate dehydrogenase, this enzyme. Do you have an idea of which ones exactly yet, and if this can tell us anything about potential treatments
it is
with supplementation?
the ketogenic amino acids that can be used to convert it to acetyl coenzyme A and used for fuel. I've discussed this with many patients, and we wouldn't recommend any special diet because it hasn't been done in clinical studies. But some patients tell me that they do better if they can reduce on the sugar and increase a few patients that I've talked to have been using a ketogenic diet and have reported some effects, improvements from that. But it's nothing that I have the data to recommend to patients.
Right.
At this stage.
Yeah. Yes.
So it's more an observation that may be more or less true. We hope that we'll see that when other groups find similar findings, I think that this is something that we should pursue further. And I think that the more we understand about the biology and the mechanisms, then the biomarker will follow.
I agree. Yes, that's a definite theme from our discussions in the last 3 days as well. I think so. Okay. Regarding the search for a biomarker, I think it's worth pointing out that one of the major objectives of the severely ill patient study was to define potential biomarkers, but also by trying to understand really more about the mechanism. The idea was to sort of collect as much data as we possibly can on this very severe cohort to really get a glimpse of what's going on in the disease. So there were, unsurprisingly, a lot of questions about this. So I'm gonna ask a couple of these, but I also welcome any comments from the rest of the panel about these types of data sets and your interpretation in your own research. So there were both Ron and Wen I think mentioned this, is the search for viruses and the fact that in this cohort we didn't actually see any major differences between the amount of viruses in the patients and in the controls. This was one of the more sort of striking findings. I wonder if you might be able to comment on that a bit more, Wen, and if it's possible that there might be viruses, that this might be because we only looked at the blood. Is it possible that they might only be detectable in other tissues, or
Yes, absolutely. I guess, Ron, you want to comment, or you want me to? So, because this is of great interest, for this particular disease, so under Ron's leadership, quite a few studies were done.
I'd like to address that a little bit, and that is the first pass was to look for virus particles in the blood, because that technology would allow us to find a new virus if there was one existing. And this was done with DeWitt up in San Francisco, and we worked with him for a number of years. We found a large number of new viruses in patients that have died from a viral infection. We've also found a live virus in a human vaccine. The nice thing about that is now there's a whole new standard of vaccine production that they have to test it for virus, and so that's not going to happen again. But there's several other ways to look for them, and a lot of people have said, well, maybe it's not in the blood. Well, as you're going to hear from Dr. Ron Tompkins, and he says that the bloodstream is really the sewer of the body, because everything gets dumped into it. And that includes when you have a viral infection, and you start to destroy some of those viruses, the DNA or the RNA gets dumped into your bloodstream. And it doesn't matter where it is. And there's been some tests for that with a virus in the brain, you find it in the bloodstream. It was a heart infection, you find it in the bloodstream. A liver infection, you find it in the bloodstream. So the technologies are really good and sensitive for doing that. Now the difficulty with that one is the fact that, and the best way to find that is with the amplification methods, and you have to target it. So you're looking specifically, is this one? And so what we did originally was really focus on a lot of the herpes viruses because there's a lot of concern that they may be a problem. And the patients have less herpes virus in DNA in their bloodstream than healthy controls. And this other thing with looking for these common viruses that are present, it's not significantly different, but the fact is that the patients have less of that virus than healthy controls. And so that also could all be just because of the immune system being somewhat activated. We did that largely because of the concern that a lot of patients feel they have constant viral infections. Now, we can't rule that out because we haven't looked at enough patients, but it also suggests that maybe they don't have a virus, they just feel like they do. And that's possibly because of the activation of the immune system. It's not in the interest of the virus to make you feel bad because you won't spread it. You'll go to bed and you won't spread it. So it's a very bad idea for the virus to do that. But it's a good idea from the point of human evolution. If you get a viral infection, make you collapse on the spot and not interact with anybody, because then it won't spread. And so I suspect a lot of that feeling that you have is just from the immune system, not from directly from the virus. So we have to know what's activating the immune system. So we're going to continue to look. So we're going to look through all the different organisms. The next one on the line is all the parasites and the trypanosomes. We have that all ready to go, and that stuff is started. We have to do the RNA viruses, and we will do that. And then we have to do the funguses and the molds, and that's a little more difficult.
Well, speaking of different organisms that are involved in the disease, there are questions about the microbiome. I wonder if you could elaborate a bit on the differences in the severely ill patient study regarding the gut microbiome. Are there more or less of certain bacteria? Do you see any sex-specific differences between males and females?
Yeah. Sure. First of all, Maureen's lab has a few other labs
that was going to be my next thing. I think
as well.
We have two people who have done a lot of this here. Right. So yes.
So just from the 20 severely ill patients that we saw, there was a difference between gender, and we don't really know at this moment what's the cause of it. And the biggest difference between males and females in these patients is verrucous microbia, and we don't really know whether that's a coincidence or it actually has certain effect on the patients. The second aspect of this was, since we see very low IPA, indopropionate, in the patients comparing to the controls, so we went to look for the bacteria that was known to generate this in the gut, and we couldn't find it. But it's only in this subset of patients. We certainly need to verify this study to see whether it's reproducible or not.
Yeah. Maureen, I don't know if you wanted to add to that based on your studies.
Well, not really. I think that our studies and the study from the Columbia group, basically shows that there's a difference in the gut microbiome between patients and controls. What effect that is happening on, what effect that has on the patients is difficult to know. It's not feasible to recommend any treatment right now. We don't know how to modify specifically the gut microbiome. There is, however, a study going on in Norway in which people will undergo fecal transplants, and it's basically a clinical trial to see if that can improve individuals' condition with the disease. And they'll also be collecting blood before and after the fecal transplant, so they can also compare what happens with other various parameters and whether the fecal transplant really is a help. I have gotten emails from patients who claim that fecal transplant helped them, but it really needs to be studied in depth and to find out if it's actually the case.
Okay, there were some questions about related diseases as well, as there are quite a few similar diseases, and certainly one of our questions in the last 3 days was what lessons we can take from diseases showing similar symptoms to MECFS. One of those, as many of you will know, is Ehlers-Danlos syndrome. It's a genetic disorder that affects collagen and connective tissue. There's been a significant overlap detected between Ehlers-Danlos and chronic fatigue syndrome patients, suggesting that there might be some similar mechanisms at play. There was a question for Wen is about the differences found in the severely ill study about the collagen building proteins, and do you think that might be related to this overlap between EDS?
Yeah, I guess it's consistent with what you said. Again, this is a relatively small study. We should,
Sure.
As Maureen showed, compare with all the published results as well as ongoing results to see whether that holds. Obviously, hydroxyproline is a significant component of proline, which is a significant part of collagen.
Yeah.
So it is consistent at the moment.
Yes. Another disease that was brought up is multiple sclerosis. As many of us know, that's probably the most similar disease to MECFS in a lot of respects. And Jonas had mentioned that you're also doing some studies on MS, and there have been some similar phenotypes. I wonder if you might comment on what this can teach us. Can these studies in MS that teach us about more about MECFS? Might it be easier to do that just because we have a bit more of a biological diagnosis for MS?
Yeah, definitely. I think first of all we can learn a lot about the history and understanding of MS, how multiple sclerosis, and how that has evolved over time, and hopefully we will see the same involvement for ME/CFS then. But also now, actually, saying from neurological point of view, I mean, MS, multiple sclerosis, is maybe the one only disease where we actually now can have some really efficient treatment and even cure to some extent. So I think that is something we should learn from. And also, as you say, we have good biomarkers for multiple sclerosis. We have imaging techniques we can use. We need to see more of that in other neurological fields, of course, but also in ME/CFS. So definitely something to follow.
Any other comments? Yeah.
There are also some studies indicating that the POTS, the postural orthostatic tachycardia syndrome, may have an autoimmune basis, which also has some overlap to ME/CFS.
Mm-hmm. All right. So there are many questions about treatments, of course, and I said we cannot talk about very specific recommendations. We can certainly talk about the prospects of all of this for treatments. We've alluded to some of these. I want to address the first group of questions as it's about stem cells, since that was related to what I presented this morning. So I want to clarify that there are no existing stem cell therapies on the market for ME/CFS that have been approved. So it is still in the research phase. We're hoping that does become the case, but that's not something that's actually been shown to work. I think it's much more important to do research first to understand what cells are being damaged in the disease, and then you might be able to try to replace those cells. But even that is very elusive right now, which is why the research that this community is doing is so important to understand. I think it's also, that's also a word of caution maybe to think about when it comes to other treatments that might be indicated by some of these findings with the metabolism. I mean, we see certain things going up and down in metabolism. That doesn't necessarily mean that we're advocating to run out and try inhibitors or supplements accordingly. This is in the research phase, so I think it's important to be cautious about that. That being said, I think what we can have a productive discussion about is the indications where things might go if we're able to validate these effectively in larger cohorts in different, using different technologies and so on. So one of the other major treatment-focused questions group was, of course, about rituximab because of Oystein's recent presentation this morning. There's a question about long-term experiences with rituximab. If you're aware that there have been anyone who has done 5 years or more with this drug, and if that might be detrimental to the immune system. And of course, if you want to say anything else about rituximab that you did not say this morning, I invite you to extend your comments.
In general, rituximab gives a reversible B-cell depletion. What we see from a single infusion is that usually after maybe 3 or 4 months, the B cells start rising again and usually back to the same levels, maybe sometime between 6 and 12 months. When you give maintenance treatment, you have mainly data from lymphoma, which I also work on in oncology work. This is a low-grade, these are different types of lymphoma, and in the low-grade lymphomas we use maintenance therapy. And there are studies with relatively often given rituximab every 2nd month or every 3rd month in lymphoma for 2 years, 3 years, 5 years, and there's been also some studies indefinitely until disease progress. So of course it perturbs the immune system by removing the B cells, and there are also important interaction between B cells and other components of the immune system. But it has been, in our clinic we use it usually for maintenance treatment for 2 years in lymphoma, and then we stop and observe the patients. But in the long-term run, if you use it maintenance for a long time, you can have low levels of immunoglobulins and could be predisposed to infections. But from 2 years treatment, you usually don't drop very much in immunoglobulin levels. So it's not a problem. And in our study, as you will see when it comes out, there's no increase in infections or antibiotic courses due to 2 years of treatment. If you treat it 7 years, you will see for sure hypogammaglobulinemia, low levels of immunoglobulins. In general.
Okay. Jonas, there were a couple of questions about your observations with pregnenolone. I hope I'm saying that correctly. And whether you believe that might lead towards a potential treatment if that can be validated in larger cohorts, if the supplementation might help with that in the long run.
Potentially, yes, but I mean, it's very hard to say in such an early stage. I would say, I also think about, the reflection of this being a signal of malfunctioning mitochondrial activity that could, of course, maybe it's the mitochondrial dysfunction we need to treat, not the steroid production as it is. So it's hard to say, but I think, the steroids are extremely potent, so of course they are involved in so many different functions in the body. So this regulated steroidenesis will lead to a lot of problems.
Yes.
I just want to add one more sentence, because in general use, the side effects from rituximab are usually mild, and it's a good tolerated drug. But all kinds of medications, also when you manipulate the immune system, can have rare side effects that can be severe for patients that we usually don't observe. But if you treat enough patients, you will sooner or later see one with another reactivation of hepatitis or some chronic infection or something else. So I think it's important that's why we should use these treatments within clinical trials to avoid so that we know if there is efficacy before we put patients at the risk for side effects. Although they are not very common, they can occur, and that's part of the balance.
Yes.
Yeah.
Yes, between the hope and the potential harm. Yes, absolutely. That's a very important point. So we're just about done, but I wanted to invite one last round of comments on what you think are the most exciting differences that have been observed between patients and controls that might be able to lead to a treatment. All of them? Yeah?
I think we'll hear some good ones this afternoon.
So we're going to pass the buck down to the afternoon speakers. That's good. Okay, well, let's revisit that in the afternoon panel. I think everybody's hungry, so let's recharge our metabolisms. Lunch is waiting for you outside. We will reconvene here at 1:45. Thank you so much to our panel and morning speakers.